Technical Veterinary Learning TVL channel, founded and produced by Professor Ahmed Mamdouh El Sharif, Professor of Epidemiology and Infectious Diseases of Animals, the ex-head of Department of Veterinary Medicine, the ex-Vice Dean of Faculty of Veterinary Medicine, Bani Suef University. We will speak today about an important part of the trip. To reach to an accurate diagnosis, you have to be aware how to collect clinical samples. Accurate diagnosis of different infectious diseases of animals is the true base of effective control to them. Diagnosis done through use of definite knowledge from one or more basic sources of information, namely are the followings. Number 1, history of the disease. Number 2, epidemiology of the disease. Number 3, clinical signs of affected animals or herds. Number 4, post-mortem findings. Number 5, results of different laboratory tests. These 5 steps have to be done to reach to an accurate diagnosis of any disease. Examination of diagnostic materials is an important function of veterinary laboratories that help different veterinarian practitioners in effective treatment and for application of different control measures against resurrection of this trouble again. Reading and interpretation of laboratory results should be done by veterinarian clinician, not by specialists in bacteriology, biochemists, pathology, and etc. Because in veterinary medicine, laboratory evidence didn't form readily and immediately evidence. It is an important step to understand the difference between diagnosis and investigation services. Diagnostic services usually done for a real and present health problem. Investigation service denote to a selected problem or problems that is studied under field and laboratory means to discover and discuss the cause, epidemiology, science, laboratory diagnosis, and control of this disease or problems. None of the disease characteristics, occurrence, and past experience have equal importance for accurate diagnosis because of clinical and pathological features of multiple diseases are similar, so accurate diagnosis could be decided by usage of different laboratory methods. The goal of most laboratory tests is to provide the base of accurate diagnosis to different infectious diseases of animals side by side with epidemiological data, clinical signs, and post-mortem features. This is a clinical examination sheet have to be done and filled by every veterinarian clinician who, when he visits a case or a herd for accurate diagnosis, as all the informations filled here will help him step by step to reach to an accurate diagnosis. This includes owner's data, which includes the date of the visit, name of the owner, the address of the owner, and phone number. Animal data, including species, sex, previous history of a disease, the temperature of the animal, the present history, have to be seen by the clinician. Clinical examination have to be done for the head, including eyes, ears, mouth, skin, sinuses. neck examination including jugular vein, esophagus, skin. Chest examination should include lymph nodes, heart, lungs, and skin. The abdominal examination should include lymph nodes, rumen, abomasum, and skin. Rectal examination should be done for seeing the actual status of the urinary bladder, large intestine, rumen, and kidneys. Pregnancy diagnosis in case of females as positive or negative. Limbs condition, tail condition, or the others. Clinical picture seen by the veterinarian clinician. This clinical examination sheet is a helpful guide for them to reach to an accurate suspect of the health problem they are invited to discover. We will speak in the following slides about the details of clinical samples collection. Knowledge about selection and collection of proper clinical specimens are important for helping any accurate diagnosis. Principally, different clinical samples from live animals, freshly dead carcasses, and or aborted materials should accompanied by the following descriptions. Number 1. Complete description of the examined animal or animals. Number 2. Time and date of death. Number 3. Time and date of specimen collection. Number 4. Examination of requirements. Number 5. Type and nature of the sample. Number 6. Expected diseases. Number 7. Mortality rate. Number 8. Morbidity rate. Number 9. Case fatality rate. Number 10. Different clinical signs that presents. Number 11. The different post-mortem signs that be found during post-mortem examination by the clinician. Collection of correct clinical samples should take in consideration the following rules. Number 1. Collection of samples should be done early during any outbreaks. Number 2. Collect the correct samples for each predicted disease. Number 3. Collection of clinical samples from right animals is very essential. Number 4. Choose the right time for samples for collection. As an example, midnight is the proper time of blood samples for filarial detection. Number 2. Respiratory affections due to viral agents. Collection of early samples help isolation of infectious agent, while late samples will be contaminated with secondary bacterial agents. Number 3. Acute and convalescent stage sera are for diagnostic value for determination of rising the antibody titers as in case of bovine viral diarrhea infection. Number 4. Clinical specimens from decomposed carcasses are unsuitable for laboratory examination such as Bacillus anthracis. Number 5. It chooses representative samples for each suspected disease as last streaks of milk for isolation of mycobacterium organisms in case of tuberculous mastitis, and an intestinal loop for detection of clostridial toxins in case of enterotoxemia. Different clinical specimens or samples. There are 16 clinical specimens could be collected from suspected animals, namely: Number 1. Blood sample. Blood smears are collected easily from animals' ear vein. Also, the whole blood samples are collected from jugular vein or mammary vein. Blood samples collected for bacteriological cultures as well as for serological examination are taken in sterile containers and tubes. Area of collection should be shared. Then disinfected with 70% alcohol. A needle number 8 is used for collection of the blood in a sterile tube for bacteriological culture or allowed to clot for serological diagnosis. The third type of blood sample, whole non-coagulated blood. The whole blood sample taken in a bottle of 100 ml in size with a 1 ml of 10% solution of potassium oxalate. This sample is used for chemical analysis of the blood. This figure showed how to collect blood from ear vein to make blood smear. Figure number 2 shown collection of the whole blood from jugular vein. Figure number 3 show you how to collect the whole blood from jugular vein. Figure number 4 shown you how to make the blood smear. Milk sample. Cleaning, drying, and disinfection by 70% of alcohol of the udder and teats should be done before collection of milk samples. The first strips of milk discarded, then second strip collected for bacteriological examination, and last strips were collected for mycobacterium species and procella isolation. Milk samples derived within 8 hours from culturing or adding of boric acid as 1 part to 4 parts of milk. Addition of boric acid didn't interfere with different tests. Figure number 5 shown you a collection of right milk sample. Figure number 6 shown you how to collect a milk sample by a teat siphon in a test tube. Resistance of Indiana vesicolovirus to physical and chemical action. Temperature. This virus inactivated by 58 centigrade for 30 minutes. The survival. Inactivated by sunlight, and it survives for long period at low temperature. Figure number 7. Sterilized catheter used for collection of urine sample for bacteriological examination. Figure number 8. Collection of a urine sample by a sterilized catheter. Sputum sample. Sputum is collected to examine suspected tuberculous animals. Sputum was examined either culturally or by direct microscopical smear. Sputum obtained from walls of stables in front of animals. Sputum samples collected on a clean paper sheet that hold in front of animals post-induction of cough. Animal-induced cough normally if it's tuberculous during the dawn of the sun or during the cold periods. Induction cough occurs through closing of nostrils of the animal until near distress of respiration. Sputum collection could be done by usage of a sputum cup. Fecal sample. Fecal specimens were collected for both bacteriological and parasitological examination as follows. Number 1, sterilized rectal swabs were collected for a bacteriological examination. Number 2, fecal sample for parasitological examination should be collected by clean glove directly from rectum into a 100 ml bottle that packed tightly to exclude air for retardation of eggs development. Number 3, in case of dead animals, abomasum and the first 15 feet of the small intestine contents are collected by ligating them and delivered to the laboratory. These contents are removed by squeezing intestine between fingers with addition to formalin with 0.5%. Figure number 9 explain by the picture the usage of a clean new glove for collection of fecal sample from rectum. Figure number 10 showing you how to collect a fecal sample directly from the rectum. Figure number 11 showing the fecal sample that taken directly from the rectum. Pus sample. Pus specimen should be collected from closed abscesses for accurate detection of the causative agents and for perfect treatment. The closed abscess cleaned, clipped, and washed by soap and water, then disinfected with 70% alcohol. Pus sample. This done by usage of lancet. Will open the abscess, then bacteriological swabs will use to collect the freshly exuded pus. Number 2. Usage of large bore needle to enter the abscess to allow dripping of pus into a sterile tube. Collected pus samples used for making either pus smears and or culture of it. Skin scraping sample. Diseased cutaneous areas. Examined carefully before collection of samples. These areas then wetted by mineral oil to prevent distribution of suspected scrapings around during the collection. Skin scrapings collected from edges of suspected areas after picking up of a cutaneous fold tightly between thumb and forefingers. Skin scrapings which adhere to the plate should be transferred to any of the following, namely a clean, dry petri dish, test tube, plastic sheet, paper envelope for culturing and or microscopical examination. This figure showed how to collect skin scrapings at neck area from edges of the lesion by planted scalpel at petri dish. Collection of skin scrapings from a neck by usage of planted scalpel. This figure showed you the skin scraping samples inside the petri dish. Hair samples. Hair specimens were collected from the edge of suspected area by usage of suitable forceps. These samples were used for culturing and or microscopical examination of these hair shafts. Tissue specimen for histopathological examination. Parts of sections from edges of a lesion containing parts of the neighboring health tissue were taken. This specimen taken as a block with 0.5 cubic centimeters. Every part put it in 10% formalin until making a pathological smears. Fetal membranes and dead fetuses sample. A part of fetal membrane that include cotyledon were collected. These parts rinsed under tap water, wrapped in a grease-proof paper, covered with cotton, and wrapped in a strong plastic sacks for delivering to the laboratory. Number 2. A complete aborted fetus should be sent in a clean dry box post-closing all natural orifices with cotton. Number 3. The stomach aborted fetus tied off at both ends and sent without preservatives with the spleen, pieces of liver, and lung to the laboratory. Semen sample. Semen specimens are collected from suspected males by usage of a sterilized artificial vagina for isolation of causative organisms. Immediately post-collection, semen sample protected against sunlight and extreme of temperature. In case of non-pressure of artificial vagina, last drops of semen that escape from the still erected penis immediately post the bull gets down after thrust should be taken. Preputal wash sample. Preputal washings are obtainable through a planted catheter 50 cm long that attached to a flutter value apparatus with a 200 ml bottle be attached with 90 centimeters of rubbering tubing. To obtain perpetual wash, the area has to be thoroughly washed with soap and water, clipping of attached hairs and disinfected with 70% alcohol. Catheter and the apparatus used have to be sterilized. Collection of perpetual washing done through the following process. Number 1. Insert the catheter into prepuce to the level of glans penis with closing of perpetual orifice by gauzing bandage. Number 2. 100 ml of sterile normal saline or nutrient, both inverted from the bottle into the prepuce, which massage to wash off the suspected material from the mucous membrane of the prepuce. Number 3. This washed fluid, sterile saline or nutrient broth allowed to come back from the prepuce by gravity to the bottle again. Vaginal mucus sample. Vaginal mucus secretions also got by usage of tampons as the following. A white tube containing tampons with loose plunger is placed as far as possible into the vagina and left for at least 20 minutes for obtaining the vaginal mucus secretion. The tampon is squeezed into a sterile McCartney bottle. Fungal specimens collection. Collection of specimen for diagnosis of suspected fungal diseases should be taken from lesions. 2 samples were taken where a part of affected tissue should be immersed in 10% normal formalin for histopathological examination, and the other parts either directly cultured in separate agar or refrigerated until culturing or laboratory animal inoculation. Viral specimens collection. Viruses infect animals shows a tropism for certain tissues. This fact needs special care during selection of specimens for making accurate diagnosis of any viral disease. Tissue sample have to be placed into a sterile wide mouth bottle and sent on ice or in 5 to 10 values of sterile 50% buffered glycerine to the laboratory. Toxicological specimens. Most of poisons could be achievable in the liver, kidney, and or stomach contents. Sample consists of stomach content in a jar and 300 to 500 grams of liver and or one or both kidneys in another jar. The clinician should do his best to find some ideas about the possible nature of the poison to help the analyst. Golden points for sampling. These golden points have to be followed for a good sample collection. The first golden point. Veterinarian clinician should remember that errors could occur during taking of any specimens, so they have to cleanse two representative sample truly. The second golden point. Errors in collection of specimens could affect on important results, as in case of parasitological detection of eggs, detection and isolation of mycobacterium organisms, contamination of milk samples with fecal matter. The third golden point, delaying in transferring of samples to the laboratory may result in error, as WPCs are reduced by 10% within 24 hours from taking of their sample. Specimens needed for laboratory diagnosis of most important of infectious diseases will be the following. In case of abortion-causing diseases, the samples will be cotyledons, fetus, fetal stomach contents, liver, lungs, and fetal membranes. In case of actinomycosis and actinobacillosis, the samples are pus swabs, pus smears, part of suspected lesion in 10% of normal formalin. In case of anthrax, specimens in case of ruminants are blood smears from ear veins directly post-death. In case of equines and pigs, smears were taken from local swellings. In both parts of ears were taken. In case of bacillary hemoglobinuria, the samples are blood samples, urine, portions of the liver. In case of blue tongue disease, samples are blood during fever, heart blood for viral isolation, bone marrow, spleen, liver, double-paired serum for serology. In case of brucellosis, the samples are serum, milk, vaginal secretions, and aborted materials. In case of cough diphtheria disease, swabs from the deep ulcers at the oropharyngeal area were taken. In case of caseous lymphadenitis, the samples are pus, serum, and affected lymph node. In case of rinderpest disease, the samples are blood during fever, saliva, lymph nodes, spleen, and bone marrow. In case of colibacillosis, the samples are intestinal loop, blood, spleen, lung, and liver. In case of CBPP, contagious bovine pleuropneumonia, sputum, nasal, and bronchial secretions, thoracic exudate of lymph nodes, and portions of affected lungs are the suitable samples have to be collected. In case of contagious ophthalmia, pink eye disease, the suitable samples are swabs and scrapings from affected conjunctiva. Suitable samples have to be collected in case of CPD, contagious pustular dermatitis, are serum, scabs, and biopsy from the contents of intestinal loop is the suitable sample of enterotoxemia disease. Suitable samples in case of epizootic lymphangitis are serum, pus from unopened lesions with addition of 500 international units of penicillin per ml of sample. In case of foot and mouth disease, the most suitable samples are blood during fever, vesicular fluid, epithelial tissues from lesions edges, lymph nodes, heart, thyroid, double-paired serum, and all samples collected and sent to labs in frozen state or in glycerol buffer with pH 7.6. In case of hemorrhagic septicemia, we have to collect the following samples: heart blood, bone marrow if animal died for more than 8 hours, nasal discharges, tracheal wash, lungs, and spleen. In case of infectious bovine rhinotracheitis, IBR, nasal discharges at early stages of the disease, swabs from nose, conjunctiva, vaginal and preputal wash, semen, spleen, brain, aborted fetus, and double-paired serum are the most suitable samples in this disease. In case of infectious foot rot, samples are serum, scrapings, or smears from ventral aspect of the foot. In case of Johne's disease or paratuberculosis, the suitable samples are fecal matter, intestinal smears, colon, ileocecal lymph nodes, and serum. Listeriosis. The most suitable samples in case of listeriosis are silage, abomasum contents, placenta, urine discharge, milk, feces, serum, liver, and the spleen of aborted fetus. Catarrhal fever or MCF. The most suitable samples are blood, buffy coat of blood, lymph nodes, spleen, and brain. In case of malignant edema or gas gangrene, the suitable samples are aspirated fluid from lesions, impression smears, bacteriological swabs from the brain, blood, and serum. Bovine viral diarrhea or mucosal disease, BVD-MD. The samples for suspected cases by this disease are from live animal, nasal discharge, ocular discharge, semen, feces, urine, and puffy coat of blood. From dead animals, placenta of aborted fetus, double-paired serum, lymph nodes, tonsils, spleen, lungs, and intestine are the most suitable samples. In case of rabies, the head of suspected animal sent on ice to the laboratory where brainstem and cerebellum was taken. Number 2. Saliva and salivary glands were sent on ice also to the laboratory for viral isolation. In case of toxoplasmosis, placenta from aborted animals, aborted fetus, brain of aborted fetus, and serum are the most suitable samples. In case of African horse sickness, before collecting or sending any samples, proper authorities should be contacted. Samples should only be sent under secure conditions and to authorized laboratories to prevent the spread of this disease. The first sample taken in case of African horse sickness disease, blood from live animals, are unclotted whole blood collected in an appropriate anticoagulant at the early febrile stage and sent at 4 centigrade to the laboratory. Samples are the following. Number 1. Blood from live animal, unclotted whole blood collected in an appropriate anticoagulant at the early febrile stage and sent at 4 centigrade to lab. The second sample in a case of African horse sickness suspicion are preferably double-paired serum samples should be taken 21 days apart and kept frozen at -20 centigrade. Preferably double-paired serum samples should be taken 21 days apart and kept frozen at -20 centigrade. The third suitable sample that have to be taken in case of suspicion of African horse sickness are spleen, lung, and lymph node that collected from freshly dead animals and placed in appropriate transportation media, then sent at 4 centigrade to the laboratory. Don't freeze the samples taken from dead animals. Spleen, lung, and lymph node samples collected from freshly dead animals are placed in appropriate transport media and sent at 4 centigrade to the lab. Don't freeze samples taken from dead animals. In case of equine encephalomyelitis disease, blood during early fever and brain samples are suitable. In case of equine infectious anemia, the most suitable sample are blood samples. In suspected case of strangles, The pus and nasal swabs are most suitable samples. Lab diagnosis will be continued. Please subscribe the channel to follow the newest videos about veterinary medicine.